Okay. Well, hi, everyone. Welcome to today's Research Spotlight talk. We are broadcasting live from the Yale Peabody Museum, which, as many of you know, is undergoing renovation. And we all look forward to inviting you back really soon in April. And my name is Katrina Damrell. I'm a Master's of Environmental Management student at the Yale School of the Environment. And I'm also the Research Spotlight Assistant at the Yale Peabody Museum. The Research Spotlight series is the Peabody's graduate student speaker series. We highlight different graduate students who do their research in connection with the museum's collections. And I have a few housekeeping details to run by everybody before we get going. So for tonight's webinar, we've disabled the chat function. So use the Q&A function, which is down in the lower right part of your screen if you have any questions. And you can submit them at any point during the webinar. My colleague David is off screen here. He's monitoring the questions for everyone. And I will say we typically receive more questions than we have time for to answer. So I apologize in advance if we don't get to your question tonight, but we'll be able to connect you to both the Peabody staff and also Mallory, who I will introduce in a second. And then finally, our program's also being live captioned. So thank you, Kristen, for captioning for us tonight. And you can turn captioning on the bottom right of your Zoom window as well. All right, so we are joined here today with Mallory Cox. Mallory is a PhD candidate at Yale's Department of Anthropology in the Council of Archaeological Studies. And she also grew up in Smyrna, Tennessee. Her work focuses on researching methods to identify hemozoin, which is a crystalline pigment that accumulates in the bone marrow of individuals infected with malaria. Her talk today will illuminate the challenges of studying evidence of disease through archaeological remains. She will share her archaeological investigation using high-powered magnets to examine human skeleton bones for malaria in tropical Americas before 1500 CE. And her novel techniques have major significance for modern-day malaria detection and diagnosis. So with that, I will pass it off to you, Mallory.
Thank you, Katrina.
Yeah. Share screen.
Yes.
Oh, I think if you just print slideshow presenter view. Yeah. And then go back to Zoom. Oh yeah, right here. Yeah, no problem. 1 second, folks. Here we go. All right.
Perfect. Good afternoon. Thank you everyone for joining, and thank you very much to Katrina for the lovely introduction, and to David Heiser and the Peabody Museum for the invitation to share with you today. As Katrina stated, my name is Mallory Cox. I'm a doctoral candidate in the Department of Anthropology in the Council of Archaeological Studies. And the research I'm sharing with you today synthesizes my dissertation work over the last 6 years. So as an archaeologist investigating health and disease in ancient populations of the Americas, my research involves analyzing and sampling human skeletal remains. In my presentation today, I have multiple slides with images of skeletal remains. And if you prefer not to view this content, consider leaving the webinar now, or maybe just adjust your screen that you can still listen to the presentation. And if you have any questions about the human remains held in the Peabody collections, including those subject to NAGPRA repatriation, please feel free to contact our NAGPRA repatriation registrar, Jesse Cohen, whose contact info will be available in the chat shortly. We'll get started. Hey. Malaria. Malaria is an infectious disease caused by parasites of the Plasmodium genus. These parasites are transmitted through the bites of infected Anopheles mosquitoes. Currently, there are 6 species of malaria infecting humans, but there are 2 primary species that account for most of the infections worldwide. Plasmodium falciparum and Plasmodium vivax are the 2 species that we're referring to. And Plasmodium falciparum is responsible for most deaths associated with malaria and the cerebral form. And Plasmodium vivax is actually much more widespread geographically where it occurs. So the heat map on the bottom right shows the current distribution of all malaria cases worldwide. And malaria remains a major global health burden, as in 2022 alone, there were over 600,000 deaths from malaria. So modern diagnosis and treatment of malaria present unique challenges that have to be overcome in the next generation of malaria control. In addition to increasing insecticide resistance and anti-malarial resistance, warming global temperatures are steadily expanding the habitat of the mosquito vector that transmits the disease. So while the gold standard for malaria diagnosis involves the identification of the parasite using a blood smear, that method is not rapid. It requires a trained microscopist and to identify the malaria at the species level. And it's just not accessible as a method for many regions of the world where they're underserved. So we can do DNA testing to look for the malaria parasite, but that is actually very expensive. It's time consuming. It requires a lot of resources. So that also makes that not much of a viable option in many parts of the world. And finally, rapid diagnostic tests. They have been the go-to for some time for diagnosing malaria. However, in recent years, we have seen that these are no longer as reliable as they were. The parasite is evolving so rapidly that it now no longer produces some of the proteins that these tests target specifically for identification. So we can have missed infections altogether, or just false negatives, for example. So archaeology and anthropology in general are very interdisciplinary studies. We look to various lines of evidence to understand how people lived in the past. And ideally, we would have written records that would explain diseases and their symptoms, in the terms that we understand them today, maybe even use the same name to call them that we do today, but that's often not the case. What is more common is that we see symptoms described and sometimes depictions of disease vectors interacting with humans. But in some places in the world, particularly where I work in the pre-contact Americas, we have less written records than we'd like. So we have to use every technique available to us to look for malaria. And in this case, we start with paleopathology. We are fortunate enough to have some skeletal remains to start our investigation. So most of the work that we do in the Macintosh lab falls all under the paleomicrobiology umbrella. So when an infected female mosquito takes a blood meal and bites a human, she actually injects that parasite into the bloodstream. And those parasites immediately localize themselves to the liver, where they can evade the immune system and hide out for a period of time in order to replicate. So after about 10 to 12 days time, the parasites will then leave the liver and invade red blood cells. And it's at this point in the stage of the infection that we start to see the symptoms. So the most characteristic symptom of malaria is the intermittent cyclic fevers. These come with severe chills. And this is usually accompanied by headache and fatigue, vomiting and diarrhea. Some of these blood stage parasites go on to sexually mature, and they move to the bloodstream and will be taken up into the next blood meal, and the transmission continues. Now, for us, we want to look closer at the blood stage of infection specifically, because this is where hemozoin is produced in large quantities. Katrina mentioned earlier in her introduction that hemozoin, the crystalline pigment that's used as a biomarker for identifying malaria in living humans, is at the center of my research. So while the parasite is inside the red blood cell, it digests hemoglobin and produces a heme crystal, which is observable as a brownish black pigment. As the parasite matures, more hemozoin accumulates. And we've also seen a lot of research in recent years that have shown that hemozoin actually sequesters to the bone marrow cavities. So that's what I'm showing you here on the far right. You can see an example of this where these authors showed that after 9 months after clearance of the malaria infection, these long bone marrow cavities were still contained a large amount of hemozoin crystal. So this is good news for archaeologists looking for malaria in skeletal remains, because ancient DNA is very scarce. And when pathogenic DNA is even rarer, and especially in some of these parts of the world where malaria is endemic and we have very humid, wet environments. So the good news is that Teddy Setzer and her group showed in 2012 that bone marrow can preserve in human remains for up to thousands of years. Now, now that we understand malaria today and how we might look for it in the past, I want to first share with you all what evidence we do have already for malaria in the Americas before 1500 CE. And while it is scarce, it's very compelling, in my opinion. One study was conducted in 2012 by Geertsen and colleagues using 155 specimens, liver and spleen specimens of South American mummies. And they were able to identify Plasmodium vivax antigens in almost 70% of these samples. It's a very strong indicator. And then secondly, we can start to think about anti-malarials in the past. So written records explain the sophisticated use of the cinchona bark by indigenous populations of Ecuador and Peru when the Jesuits arrived in 1500. Let me take a step back. The two antimalarials that we use today are quinine and artemisinin. Quinine is derived from the cinchona tree bark. So despite Jesuit claims to having discovered this remedy for the fevers after they arrived in the Americas, we do know that local communities introduced the use of the bark to them, and then they took that knowledge back to Europe. Because my research focuses on a time and a place in the world without extensive written records, but we do have human skeletal remains. Paleopathology is our first avenue for investigating malaria in the archaeological record here. And thankfully, we have one very strong study done by Nicole Smith-Guzman in 2015. She used a medical collection of skeletal remains from a malaria endemic country and recorded the lesions that she observed on the bones. So that data set was then compared to an outside collection of individuals who lived in a malaria-free environment. Using statistical analysis, she was able to show that there were 5 skeletal lesions that occurred most frequently in individuals from the malarious population. So that included product hyperostosis, which is the pitting on the cranial vault that you see in the far left, and also femoral cribra, which is a pitting underneath the head of the femur, on the neck underneath the head. And then finally, spinal porosity. Spinal porosity was present in nearly 90% of individuals living and dying in a malaria endemic area. So using this information, I was able to find a skeletal collection from the ancient Caribbean, which displayed similar lesions in very high frequencies, which I'm going to tell you more about now. So my dissertation research includes 105 samples representing 49 individuals from 12 archaeological sites across the countries of Panama and Puerto Rico. For each individual, we prioritize individuals for sampling if they displayed malaria-associated lesions, and also if there was more than one long bone present in reasonable condition. So for every individual, there's two long bones sampled with a total of about 100 grams, 1 gram of bone material in total. And on the right, you have actually an example of one of the individuals that's included in this study. And that young man, we believe, had severe femoral cribra. So we took that as an indicator that he should be included in this study. So here is an example of some of the malaria-associated lesions that I just mentioned in the Panamanian remains. On the far left, you see the femoral cribra. And then in the following two pictures, you can see really clear evidence of kind of a severe case of spinal porosity. So I can assure you that these are abnormal for a healthy child. This child was about 3 to 4 years old when they died. So this is an example of some of the collections that we did, some of the individuals we decided to sample for the study. So the 49 individuals come from 12 different archaeological sites. And what this timeline depicts is the span of time that each of the sites represents and was occupied. For the country of Puerto Rico, most of the collections are awaiting new C14 dates, but the cultural remains and the dates we do have suggest that those individuals were living on the island around 400 BCE to 900 CE. And for the 30 individuals sampled from Panama, the earliest come from the site of Cerro Mangote. This site was occupied continuously for over 6,000 years. And the human interments have been dated to around 2500 BCE. This is one of the earliest plant and animal domestication sites in the Americas. So really interested to see what we find there. And within Yale's Peabody Museum's Caribbean collection, the subset of skeletal remains from the island of Puerto Rico were selected specifically because they displayed great preservation. They represent various ecological sites across the island, from coastal sites to inland sites to others along rivers. And also the Yale archaeologist, Dr. Irving Rouse, who excavated these remains in the 1930s, he did a very excellent job of taking nice notes and creating a lot of drawings and images of the individual burials, so we can account for the context in a way that's not possible for some of these other places. Now, we're going back to hemozoan. This is the star of the show. The magnetic properties of hemozoan, based on the iron atoms in the crystal, have been used for decades now to isolate infected red blood cells from healthy red blood cells. So the figure I'm showing here kind of explains why that's possible. This was a foundational publication by Hackett and colleagues, 2008, where they showed that during parasite maturation, as hemoglobin is digested and heme is turned into a heme crystal, the magnetic susceptibility of the red blood cell increases in a linear fashion as the parasite matures. So magnetic susceptibility is a very standard measurement that we take in magnetometry, and it essentially tells us how strong is a material's magnetic components in response to an applied field. So that publication from the previous slide, along with this groundbreaking paper here on the left, really lie at the core of our rationale for the research we're doing. So the paper on the left was published by Gregory Nolan and David Sullivan in like 2003, and they showed that hemozoan crystals were uniquely shaped and sized at the species level. And then there was some follow-up work that showed that this is easily quantifiable using microscopy, and that the difference between some of these crystals, so the difference between species essentially, is anywhere from 50 nanometers to 300 nanometers. Differentiating crystal size with a resolution down to 10 nanometers is well within the capabilities of the instruments that we use in the archaeomagnetism lab. So we were optimistic about exploring this a little further. And these images here show kind of step by step the process that we go about, where we collect bone marrow cavity samples from the pictures on the left. And then we do a lot of imaging of those samples. In the middle picture, you can kind of see what an example of a sample looks like. It's the scrapings of bone from the cavity. For the magnetometry measurements, we then prepare the sample in a specific type of resin that's essentially invisible to our instrument. And then finally, we put them on the AGM, the alternating gradient magnetometer, which you see on the right. And we do a handful of different types of measurements to see what type of magnetic materials are in the sample and where we can go next. So the graphs I'm showing here are actually the output of a series of measurements that we take when we're assessing magnetic susceptibility. We were very excited to see that just upon first glance that the two species of malaria seem to be behaving differently in this applied magnetic field. We weren't sure yet if this was just related to the sample size or actual differences in the crystal size. So we decided to take it one step further and look at magnetic coercivity. And magnetic coercivity can be easily understood as how strong of a negative field is required to prevent a material from magnetizing in an applied field. So the figure on the left here is kind of a visual depiction of how crystallite size correlates with magnetic coercivity. So this is the rationale behind using this particular measurement. So the difference between this measurement and susceptibility is that this is actually much more sensitive. It relates to the composition of the crystal. And the chart on the right shows some of our very first preliminary measurements of the clinical hemozoan samples from various species that we're able to obtain. And we took these measurements, and we were actually pleasantly surprised to see that they all kind of hovered around 8 millitesla as a coercivity measurement. While differences in coercivity here seem very small, we do think that they're probably big enough to be statistically significant and potentially used as indicators, measurement proxies for diagnosis. So we're going to continue this work. We're going to continue working with our collaborators on the medical campus and obtaining more species of malarial hemozoans, perhaps from different stages of the life cycle infection, different size samples, and continue these studies so that we can see just how applicable our method is to hemozoan identification, both in living and ancient individuals. We started this work just wanting to know more about how to identify malaria in the past, but we did not realize that there would be any potential for contributing towards diagnosing malaria in the future. So for this, we're very excited and grateful for all our collaborators. We're working with Yale's medical campus and the School of Public Health. And this semester, we will start working with engineers to look at the possibility of miniaturizing this measurement in a small device. But with that, I'm going to go ahead and take any questions that you have. I know we only have so much time, so I encourage anyone who doesn't get their question answered today to reach out to me via email. I'm always available. But last but not least, I have many, many thank yous. Again, thank you to the Peabody Museum for this exciting opportunity to share. And also thank you to Roderick McIntosh, my primary advisor, who supported me all along the way. Thank you to Mike Carolla, my partner in the lab. Big thank you to Philip DeBlasi and Anna Brown-Ribeiro as my primary thesis advisors who encouraged me to take on the malaria project. And I won't be able to go through the whole list, but there are many, many people to thank that were major, important contributors to this work. So thank you very much for your attention, and I'm happy to answer any and all the questions that you have.
Hey, thank you, Mallory. That was fascinating and completely new set of information that I'm completely new to this, so this is helpful to learn about magnetism and malaria and diagnosing this. So we do have a few questions coming in here. So Mina is asking, is there a form of either sickle cell anemia or, and I excuse myself for pronunciation, thalassemia or something similar in the native populations of the Americas?
So this is a really good question. And there isn't actually much evidence for any type of red blood cell. So first of all, what she's referring to here is actually these genetic conditions or disorders that actually confer protection against malaria. So there's a large part of the African continent, for example, where individuals all have sickle cell trait, and this protects them from malaria. So that shows us how malaria has been a force, an adaptive pressure in that part of the world for a very long time. When we look to the Americas, it's less clear. There's no distinct skeletal lesions and obvious markings of any blood disorders, but there are some suggestions of a condition called G6PD. It's another blood condition. There's a little bit of it in the Mediterranean. But no, we don't have those markings in the Americas. It would be great if we did. But we also wonder how many thousands of years does it take, right, of exposure to that pathogen for those lesions to develop? So maybe we just haven't had the presence long enough to result in something like that we could see so easily.
Yeah, fascinating.
Thank you for the question.
Yeah. Niels is asking, did malaria spread follow the migration of humans around the world, for example, to the Americas, or was it already present? And that was actually similar to what I was just thinking, listening to you answer your questions.
Yeah, so from what we know, it does seem as though malaria went with hominins and then the Homo species around the world. Some of the most ancient forms of the disease, we believe, originated on the African continent in non-human primates. They moved over to humans a long time ago. We do think that as humans moved to Southeast Asia and about the globe, including over to the Americas, much later, that they likely brought Plasmodium with them. As far as in the Americas, it's interesting because Plasmodium vivax, the American strain, the current American strain, actually appears to be genetically distinct from its ancestor species of vivax in Southeast Asia. So what that suggests is that it has been here isolated somewhat and evolving for probably a lot longer than 500 years, which is roughly our cutoff point for contact with European colonizers. However, falciparum malaria in the Americas, it is almost identical to the West African strain of falciparum malaria. Which supports the conclusion that falciparum came with the transatlantic slave trade, but vivax was likely here much, much earlier. And so that study that Gertsen and colleagues conducted in 2012, that also supported that conclusion. They showed that while 70% of these specimens had antibodies for Plasmodium vivax, none of them had antibodies for Plasmodium falciparum. So
it's fascinating. Yeah.
That's what we think.
Edwin is asking, are there any in situ methods for identifying the presence of hemozoin in bones? Could you use MRI or NMR?
Yeah, so these are great questions. These are the types of ways that we're trying to think right now in order to develop, a portable diagnostic instrument, essentially. We can see with MRIs and other types of imaging, you can see the dark colored pigment discoloration in the marrow cavities. So it's there and it's distinguishable. But we don't yet have a way to do it in situ. There is an instrument where they're able to identify hemozoin in the bloodstream, but it's using a slightly different methodology than what we're working on. So I think we're almost there, where we can identify malaria in the bloodstream including using the hemozoin biomarker. We just can't yet do it at the level where we can differentiate between species in one test. Right. So, yeah.
Interesting. Between the species of Plasmodium.
Exactly.
Yeah.
Between the species of Plasmodium.
Huh. Interesting. Nina is asking, your methodology is nothing short of groundbreaking. When do you expect to publish? And wow, let's start with the question.
Well, thank you very much. I'm working on this publication as we speak. Working with some individuals here at Yale on the intellectual property rights and otherwise, but thank you very much for that compliment. We are very excited about this preliminary data and moving forward, and I would say that later on this year we will have publication
Fantastic.
submitted and published.
Yay. Yeah. Natasha would like to know, in the graph you showed, is the increase in magnetic property of hemozoin due to an increased amount in cells or due to maturation / increased complexity of the crystals over time?
Okay, so that's a great question. What it relates to, so first of all, the red blood cell is comprised of about 90% hemoglobin. So the majority of what the parasite is digesting while it's in the red blood cell is all hemoglobin. And what happens is the heme fragment is cleaved from the globin. And with the heme by itself, it's likely to oxidize and kill the parasite. So what happens is the parasite has evolved this mechanism where it accumulates these heme fragments into these crystalline into crystals.
Oh, okay.
Right. So the heme fragment is turned into a crystal. And it's actually the point at which the heme is stacked into the crystal that the magnetic susceptibility increases.
Wow.
So the iron in the heme molecule goes from a Fe2 plus to an Fe3.
Okay.
And that shift is where we see the magnetic susceptibility change. The crystal itself behaves differently because the interactions between the ions within it are different than the interactions between iron and hemoglobin or free heme, for example.
Fascinating.
Hopefully that was clear.
Yeah, no, that helps me too. That was a great question. Ralph would like to know, how do you see your research impacting on possible diagnostic or treatment strategies for current day malaria?
So most of the modern recent methods for diagnosing malaria all target hemozoin as an identification biomarker. Okay. So we think we're in the right space there.
Yeah,
first
it's familiar.
Of all. Yeah, it's very familiar. We know that this biomarker is reliable. It's sensitive, right? It's used every single day to diagnose malaria and treat people effectively. Another important thing, though, is that antimalarials themselves, as pharmaceuticals, they target the formation of the hemozoin crystal. So the more that we understand about how this crystal forms, how this crystal breaks down, how this crystal changes throughout the life cycle, the way that the immune system responds, and ways that we can interrupt the formation of the crystal, the more that we can contribute towards that, the more likely we are to have anti-malarial development in the future that can combat these kind of evolving parasite, as we see so much anti-malarial resistance to the treatments we use currently.
Okay.
So we're hopeful that just the hemozoin studies in themselves are going to shed a lot of light on that whole process, but then also that our method is potentially more sensitive and specific than some of the others that are available right now. To our knowledge, there's not an instrument in the world right now that can differentiate between species of malaria in one measurement. There's several people working on it, and but we're hopeful that this will be the game changer, so we can combine susceptibility and coercivity and a couple other tricks we got in our bag, and maybe nail it that way. So,
wow, that's so exciting. Yeah,
thank you.
Yeah. Philip would like to know, what is the actual mechanism of bone destruction is it by the plasmodium?
So in the bone marrow cavities, we have seen that hemozoan deposition does lead to bone loss. If that's what you're referring to, I think that might be. We're not entirely sure if it is just an overactive response of the immune system and bone being broken down at a rate that is not commensurate to what's being built, or if it also relates to this condition in the bone marrow specifically, where the bone marrow is kind of in overdrive constantly. As the parasite digests your hemoglobin, and you quickly become anemic, and the body goes into overdrive and trying to produce new red blood cells. Well, the red blood cells are produced in the bone marrow.
Okay.
So we believe that there's like this kind of complex relationship there between the parasites maturing in the bone marrow, first of all, and then the hemozoan that is deposited and left behind by them, and damage that might occur to bone as it is trying to regenerate itself. Yeah, and there's still a lot to be learned actually about those processes and stuff, but there's definitely a strong, clear correlation between the presence of malaria hemozoan in the bone marrow cavities and bone loss.
It's fascinating. Bonnie is asking, can you distinguish between hemozoan deposited by plasmodium and the hemozoan produced by other blood-feeding parasites, such as I'm going to try to pronounce this, schizostoma.
I got you covered.
Okay.
Schistosomes.
Okay, that was not even close.
No, it's okay. It's perfectly okay. And that's just how I pronounce it.
Okay.
So that's correct. Yes, so this is also a great question, and we didn't have time to get to it in the spotlight today, but it's very important actually, because if there are other blood feeding organisms that produce this crystal, then how can we be for sure that we're not identifying them, right? Instead of malaria. And what happens is each of these crystals, like plasmodium, plasmodium has unique shapes and size between the species. Well, we're fortunate in that schistosomes and a few other parasites that don't actually infect humans, but we know they produce hemozoan. So the crystals from each of these different diseases and species are distinct. They're absolutely distinct. So schistosomes, for example, we do have one individual where we think we saw some schistosome hemozoan crystals just based upon the size and shape. They're identical to what the literature shows, but we actually are working now with some new collaborators to try to get some schistosomal hemozoan materials to incorporate into our reference database so that we can control for that potential, and including in the Puerto Rican remains. Schistosomiasis was present in Puerto Rico and was a problem for quite some time. So it would be reasonable to expect that we might find that in those remains.
Wow. Okay, and I know we're starting to get close to the end of time, so I'm going to go through a few more questions. But if we don't get to your exact question, we will connect you guys to Mallory. Allison is asking, is treatment for the different malaria types different, so that they need to see whether Vivax or falciparum, sorry again, to see whether Vivax or falciparum, apart from one being more lethal? And I'm not sure if I read that correctly.
Yeah, no, I totally understand the question.
Okay.
It's a great one. And yes, there's relevancy there. So Quinine and artemisinin and their derivatives. This
Right.
goes beyond my expertise, so I don't get too much in the pharmaceutical aspect, but I do know that quinine and artemisinin and forms of these compounds are essentially used to treat malaria, and each one is used for a different species.
Oh,
So
I see. Okay.
it is important to differentiate between the species of malaria so that you can get the right anti-malarial treatment, but also because not only can we expect that falciparum may bring about a much worse case and potentially kill you, but Plasmodium vivax has a unique latent stage where it hides out in the liver.
Right. Is that the
For
2-week incubator?
No, actually, it's another evolutionary mechanism that
Oh,
it's
no.
evolved. So Plasmodium vivax can actually reside kind of silently in the bone, or excuse me, in the bone marrow likely, but in the liver for many months, if not years, past the clearance of an infection.
Wow.
Right. So Dr. Joe Vinetz over on the medical campus, he's one of my advisors, and he was able to see in a woman who had been in an island 71 years before
No.
and then was diagnosed with Plasmodium malariae here in the States.
No.
And I kid you not, they couldn't figure it out because they said, well, it can't be, she's been right here all this time. It's not gonna be malaria. But she had all the symptoms. She even had an
Wow.
enlarged spleen at that point.
Wow.
And sure
Yeah.
enough, that was just a really compelling example of why, we can't dismiss Vivax. It can be in an individual throughout a lifetime.
Wow.
So you want to know that you have that, and you want to
Yeah,
get the right
absolutely.
treatment to clear it from the liver.
Right.
Yeah.
And be able to have that testing be
Yeah, and have the
accurate.
forethought so that
Yeah.
you continue to monitor yourself, right, for
Yeah.
symptoms and things like that.
Wow. Wow. Oh wow, this is awesome. I feel like you have a whole packet of questions coming in. But I think we have time for probably 2 more. See if we can get through 2. So Diane is asking, for clinical use, is it necessary to distinguish between the species? Would diagnosis of the parasites be sufficient to initiate treatment, which would be the same no matter which whatever the species is?
So the treatments are slightly different. Again, this goes a little bit beyond my expertise.
Right, like the quinine
with the medical field. I do know that there are different anti-malarial treatments that are prescribed depending upon which species you have.
Mhm.
So whether it's a blood smear, where a microscopist identifies the species level infection, or whether it's a rapid diagnostic test,
Yeah,
where it targets specific antigens, there is a lot of value in differentiating between them for the purposes of treatment. Yes, absolutely.
that's awesome. Okay, and I think this will be the last question. Oh wow, there's okay, it's hard to pick, but Danielle is asking, thank you for your presentation, first of all. You mentioned that parodic hyperostosis is one of the markers of malaria, but did you consider other diseases or factors as a cause for this symptom? For example, Bartonello's in the Northern Andes has a similar clinical consequence as hemolytic anemia or malaria.
Yeah, that's thank you for that question. It's a wonderful question.
Yeah.
So the issue with parodic hyperostosis and cribra orbitalia both. So cribra orbitalia is kind of the same expression as these cribrosities in the crania, but it's actually in the eye orbit. So we have parodic hyperostosis on the surface of the skull, and then cribra orbitalia in the eye orbit. And together, they seem to be, it's not entirely clear what they are diagnostic of. We do think that there is a really good likelihood that it is anemia. But then you have examples like this where you have strong evidence for a particular infectious disease where these individuals do have that lesion. So I think it is important to do a differential diagnosis and to be able to account for any other potential contributing conditions or illnesses. In the case of malaria, I think that if we use parodic hyperostosis alongside some of the other indicators that Smith-Guzman was able to identify, so the femoral porosity, the humeral porosity, and the spinal porosity, if we use those collectively, she was able to show that statistically that it seems to be strongly correlated with malaria. But it is very important to consider that there are other causes of that particular lesion. So that's a great question.
Yeah. Awesome. Well, yeah, awesome questions this evening, everyone out there. Thank you for joining us. This is such a fascinating presentation, Mallory. I'm so excited to hear about your next steps, especially in regards to applying this in modern day settings. It's very exciting to hear about your journey in that regard.
Thank you.
Yeah, yeah. And so for the audience tonight, if you have enjoyed this talk and want to revisit it or share with your friends and family, we will be uploading this to the YouTube channel for the Yale Peabody Museum. So keep checking there for that. It'll be up there. It might take a few weeks or so, but and then the Research Spotlight series itself will continue on February 15th. So look out for that. The next speaker is Alex Bertocchi from the Department of Anthropology. And he will show us how the chemical composition of fossil herbivore teeth can reconstruct Malawi's ancient climate 20,000 years ago. So yeah, it's been such a fascinating series to be part of because of all the different research, and tonight's talk was just really, really special. Thank you, Mallory.
Thank you.
And yeah, please follow us on social media. And if you go on Yale's Peabody's website, we also have a link to sign up for our newsletter, which we also advertise the Spotlight speaker series there as well. And thank you everyone for joining us. Thanks to David. Thank you, Kristen. See you guys soon.
Thanks.